The energy released is used to drive H plus ions, protons, across the membrane, from the inner mitochondrial compartment to the outside. This generates a transmembrane gradient of H plus ions that serves as a source of energy, like a battery, that can be tapped to drive a variety of energy-requiring reactions. See Chapter 12. In mitochondria, the most prominent of these reactions is the phosphorylation of ADP to generate ATP. At the end of the transport chain, the electrons are added to molecules of O2 that have diffused into the mitochondrion. The resulting reduced O2 molecules simultaneously combine with protons H+ from the surrounding solution to produce water. The electrons have now reached their lowest energy level, and all the available energy has been extracted from the food molecule being oxidized. The oxygen-requiring generation of ATP is termed oxidative phosphorylation. Figure 13:18. Oxidative phosphorylation occurs in the mitochondria of eukaryotic cells and in the plasma membrane of aerobic bacteria, and it is one of the most remarkable achievements of cellular evolution. As such, it will be a central topic of Chapter 14. In total, the complete oxidation of a molecule of glucose to H2O and CO2 produces about 30 molecules of ATP. For an explanation of exactly where these ATPs come from, see Table 14.1, page 465. In contrast, only 2 molecules of ATP are produced per molecule of glucose by glycolysis alone. Regulation of metabolism. A cell is an intricate chemical machine, and our discussion of metabolism, with a focus on glycolysis and the citric acid cycle, has addressed only a tiny fraction of the many enzymatic reactions that can take place in a cell at any time. Figure 13:19. For all these pathways to work together smoothly, as is required to allow the cell to survive and to respond to its environment, the choice of which pathway each metabolite will follow must be carefully regulated at every branch point. Many sets of reactions need to be carefully controlled. For example, to maintain order within their cells, all organisms need to constantly replenish their ATP pools through sugar or fat oxidation. See Figure 13:10. Yet animals have only periodic access to food, and plants need to survive overnight without sunlight, when they are unable to produce sugar through photosynthesis. Plants and animals have evolved several ways to get around this problem. One is to synthesize food reserves in time of plenty that can be later consumed when other energy sources are scarce. Thus, a cell must control whether key metabolites will be routed into anabolic or catabolic pathways. In other words, whether they will be commissioned to build other molecules or burned to provide immediate energy. In this section, we discuss how cells regulate the intricate interconnected pathways that form the basis of metabolism. Catabolic and anabolic reactions are organized and regulated. All the reactions shown in Figure 13:19 occur in a cell that is less than 0.1 millimeters in diameter, and each step requires a different enzyme. To add to the complications, the same molecule is often a part of many different pathways. Pyruvate, for example, is a substrate for half a dozen or more different enzymes, each of which modifies it chemically in a different way. As we have already seen, Pyruvate dehydrogenase converts pyruvate to acetyl-CoA, and lactate dehydrogenase converts it to lactate. A third enzyme changes pyruvate to oxaloacetate, a fourth to the amino acid alanine, and so on. All these different pathways compete for the same pyruvate molecule, and similar competitions for thousands of other small molecules go on at the same time. One might think that the whole system would be so finely balanced that any minor upset, such as a temporary mood change in food intake, would be disastrous. In fact, the metabolic balance of a cell is amazingly stable. Whenever the balance is perturbed, the cell reacts so as to restore the initial state. Cells can adapt and continue to function during starvation or disease. This resilience is made possible by an elaborate network of control mechanisms that act on enzymes to regulate and coordinate the rates of the many metabolic reactions in a cell. As we saw in Chapter 4, the activity of enzymes can be controlled in many different ways. Many proteins are switched on or off by covalent modification, such as the addition or removal of a phosphate group. See Figure 438. Alternatively, their activities can be controlled by the binding of a small regulatory molecule, often a metabolite, to an allosteric enzyme. See pages 151 to 152. Such regulation can be positive, enhancing the activity of the enzyme, or negative, inhibiting it. As we see next, both types of regulation, positive and negative, control the activity of key enzymes involved in glycolysis. Feedback regulation allows cells to switch from glucose degradation to glucose biosynthesis. The body needs a continuous supply of glucose to meet its metabolic needs. For example, brain cells depend almost completely on glucose for respiration. During periods without food and during hard physical exercise, the glucose in the blood is used up faster than it is being replaced from food. One way to replenish blood glucose is to synthesize it from small, non-carbohydrate organic molecules such as lactate, pyruvate, or amino acids in a process called gluconeogenesis. An intricate pattern of feedback regulation enables cells to switch from breaking down glucose through glycolysis to synthesizing it through gluconeogenesis. Most of the reactions involved in the breakdown of glucose to pyruvate are readily reversible. However, 3 of the reactions, steps 1, 3, and 10 in panel 13.1, are effectively irreversible. In fact, it is the large negative free energy change that occurs in these reactions that normally drives the breakdown of glucose. For the pathway to go in the opposite direction, to make glucose from pyruvate, these 3 reactions must be bypassed. This detour is achieved by substituting a set of alternative enzyme-catalyzed bypass reactions that require an input of chemical energy. Reactions A, B, C, and D in Figure 1320. The reactions that synthesize a molecule of glucose in gluconeogenesis thus require the hydrolysis of 4 ATP and 2 GTP molecules, compared with the overall generation of 2 molecules of ATP for each molecule of glucose consumed during glycolysis. In humans and other mammals, gluconeogenesis occurs mainly in liver cells, which can keep the blood supplied with glucose by using many different molecules as the starting point. One common input is lactate. This molecule produced by overworked muscle cells is taken up by the liver, where it gets converted back into glucose to replenish depleted muscles. The balance between glycolysis and gluconeogenesis must be highly regulated, so that glucose is broken down rapidly when energy reserves run low, but is synthesized and exported to other tissues when the liver cell has sufficient energy reserves in the form of pyruvate, citrate, or ATP. If both the forward and reverse reactions in Figure 1320 were allowed to proceed without restraint, they would shuttle metabolites backward and forward in futile cycles that would consume large amounts of energy and generate heat for no purpose. One of the key control points in the breakdown of glucose lies in step 3 of glycolysis, the production of fructose 1,6-bisphosphate by the enzyme Phosphofructokinase. This is one of the reactions that must be bypassed in gluconeogenesis. See step 3 in Figure 1320 or in panel 13.1, page 430. Phosphofructokinase is allosterically activated by AMP, ADP, and inorganic phosphate, the byproducts of ATP hydrolysis. It is allosterically inhibited by ATP, citrate, and alternative fuels for respiration, such as fatty acids, which can be liberated from stored fat when glucose is not available. Thus, when energy reserves are low and the products of ATP hydrolysis accumulate, phosphofructokinase is activated and glycolysis proceeds. On the other hand, when ATP or fuel sources represented by citrate and fatty acids are abundant, phosphofructokinase is turned off, favoring gluconeogenesis and ultimately the storage of food molecules. To add an additional level of control, the enzyme that catalyzes the reverse reaction, fructose 1,6-bisphosphatase, see bypass reaction B in Figure 1320, is regulated by the same molecules, but in the opposite direction. Thus, this enzyme is activated when phosphofructokinase is turned off. Such coordinated regulatory mechanisms allow a cell to respond rapidly to changing environmental conditions and to adjust its metabolism accordingly. Cells store food molecules in special reservoirs to prepare for periods of need. As we have seen, gluconeogenesis is a costly process, requiring substantial amounts of energy from the hydrolysis of ATP and GTP. Thus, it cannot operate indefinitely. To compensate for long periods when food is unavailable, animals store food reserves within their cells. Glucose is stored as the subunits of the large branched polysaccharide glycogen, which is present as small granules in the cytoplasm of many cells, mainly liver and muscle. Figure 1321. See also panel 2.3, pages 68 to 69. The synthesis and degradation of glycogen occur by quite separate metabolic pathways, which can be rapidly and coordinately regulated according to need. When more ATP is needed than can be generated from food molecules taken in from the bloodstream, cells break down glycogen in a reaction that produces glucose 1-phosphate, which is then converted to the glucose 6-phosphate that feeds into the glycolytic pathway. The glycogen synthetic and degradative pathways are coordinated by enzymes in each pathway that are allosterically regulated by glucose 6-phosphate, but in opposite directions. Glycogen synthase in the synthetic pathway is activated by glucose 6-phosphate, whereas the glycogen phosphorylase that catalyzes the breakdown of glycogen is inhibited by both glucose-6-phosphate and ATP. This regulation helps to prevent the breakdown of glycogen when ATP is plentiful and favors its synthesis when glucose-6-phosphate concentration is high. The balance between glycogen synthesis and breakdown is also regulated by intracellular signaling pathways that are controlled by the hormones insulin, adrenaline, and glucagon. See Table 16.1, page 535, and Figure 1623, page 550. Quantitatively, fat is a far more important storage material than glycogen, in part because the oxidation of a gram of fat releases about twice as much energy as the oxidation of a gram of glycogen. Moreover, glycogen binds a great deal of water, producing a six-fold difference in the actual mass of glycogen required to store the amount of energy as fat. An average adult human stores enough glycogen for only about a day of normal activity, but enough fat to last nearly a month. If our main fuel reserves had to be carried as glycogen instead of fat, body weight would need to be increased by an average of about 60 pounds, nearly 30 kilograms. Most of our fat is stored as droplets of water-insoluble triacylglycerols in specialized adipose tissue. Figure 1322 and see Panel 2.4, pages 70 to 71. In response to hormonal signals, fatty acids can be released from these depots into the bloodstream for other cells to use as required. Such a need arises after a period of not eating. Even a normal overnight fast results in the mobilization of fat. In the morning, most of the acetyl-CoA that enters the citric acid cycle is derived from fatty acids rather than from glucose. After a meal, however, most of the acetyl-CoA entering the citric acid cycle comes from glucose derived from food, and any excess glucose is used to replenish depleted glycogen stores or to synthesize fats. Although animal cells can readily convert sugars to fats, they cannot convert fatty acids to sugars. The food reserves in both animals and plants form a vital part of the human diet. Plants convert some of the sugars that they make through photosynthesis during daylight into fats and into starch, a branched polymer of glucose very similar to the glycogen of animals. The fats in plants are triacylglycerols, just like the fats in animals, and they differ only in the types of fatty acids that predominate. The embryos inside plant seeds must live on stored food reserves for a long time, until they germinate to produce leaves that can harvest the energy in sunlight. The embryo uses these stores as sources of energy and of small molecules to build cell walls and synthesize many biological molecules as it develops. For this reason, the seeds of plants often contain especially large amounts of fats and starch, which make them a major food source for animals, including ourselves. Figure 1323. Germinating seeds convert the stored fat and starch into glucose as needed. In plants, fats and starch are both stored in the chloroplast, a specialized organelle that carries out photosynthesis in plant cells. Figure 1324. There, they serve as food reservoirs that are mobilized to produce ATP during periods of darkness. Essential concepts. Glucose and other food molecules are broken down by controlled stepwise oxidation to provide useful chemical energy in the form of the activated carriers ATP and NADH. Sugars derived from food are broken down by distinct sets of reactions: glycolysis, which occurs in the cytosol, the citric acid cycle in the mitochondrial matrix, and oxidative phosphorylation in the inner mitochondrial membrane. The reactions of glycolysis degrade the 6-carbon sugar glucose to 2 molecules of the 3-carbon sugar pyruvate, producing a relatively small amount of ATP and NADH. In the presence of oxygen, pyruvate is converted to acetyl-CoA plus CO2. The citric acid cycle then converts the acetyl group in acetyl-CoA to CO2 and H2O. Much of the energy released in these oxidation reactions is stored as high-energy electrons in the activated carriers NADH and FADH2. In eukaryotic cells, all these reactions occur in mitochondria. The other major energy source in foods is fat. The fatty acids produced from the digestion of fats are imported into mitochondria and converted to acetyl-CoA molecules. These acetyl-CoA molecules are then further oxidized through the citric acid cycle, producing NADH and FADH2, just like the acetyl-CoA derived from pyruvate. NADH and FADH2 pass their high-energy electrons to an electron transport chain in the inner mitochondrial membrane, where a series of electron transfers is used to drive the formation of ATP. Most of the energy captured during the breakdown of food molecules is harvested during this process of oxidative phosphorylation, described in detail in Chapter 14. The food we eat is not only a source of metabolic energy, but also of raw materials for biosynthesis. Many intermediates of glycolysis and the citric acid cycle are starting points for pathways that lead to the synthesis of proteins, nucleic acids, and the many other specialized molecules of the cell. The thousands of different reactions carried out simultaneously by a cell are closely coordinated, enabling the cell to adapt and continue to function under a wide range of external conditions. During periods when food is scarce, regulation of the activities of a few key enzymes allows the cell to switch from glucose breakdown to glucose biosynthesis, gluconeogenesis. Cells store food molecules in special reserves. Glucose subunits are stored as glycogen in animals and as starch in plants. Both animals and plants store fatty acids as fats. The food reserves stored by plants are major sources of food for animals, including humans.